So just a couple of words about the next speaker, Howard Stone, who is speaking from Princeton. He's at Princeton, where he leads an enormous group of 20 young research workers, or even more. He was an editor of George Batchelor's Journal of Fluid Mechanics for over 10 years. He is the first recipient of the Batchelor Prize. I could say a lot more, but it's a waste of everyone's time. So let's hear Howard.
Thank you, John, for being a kind introduction. And thank you for the organizers for the chance to speak. It's really an honor to speak in a symposium like this, recognizing and allowing us to think about George Batchelor. Before I start, I should say that my research group is really a wonderful community for me. It's an intellectual environment that I find extremely healthy, and I just wanted to recognize them at the start because the different things that I tell you today, if they were successful, it's due to my research group. And this photo was taken shortly before the shutdown happened just over a year ago. Okay, so my connection to George Batchelor, just by way of introduction, I was a student of Gary Leal. Gary was a student of Andy Acrivos, and Gary was a postdoc with George Batchelor. I was fortunate to do a postdoc in DAMP with John Hinch, who was a student of George's. And that was also an introduction for me to DAMP. So I occasionally met George at tea time. I'm not gonna share with you the story where I spilled tea all over his carefully written notes, which you can imagine was a disaster for a young scholar. And I served for 10 years as an associate editor of JFM, the first part of that when George was an editor. And I think it's, I should certainly say I've always been a big fan of DAMP. You've heard it was founded by George, and it remains a leader in education and research, and that's special to me and many around the world. Okay, so where to begin? So I thought I'd begin with the student and the mentor. So here's a picture of George Batchelor on the right and his mentor, G.I. Taylor, on the left at George's graduation day. And I just want to point out that the first conference I was ever able to attend as a student was to listen, and it was Fluid Mechanics in the Spirit of G.I. Taylor. And that was actually 35 years ago last week. So it was really a wonderful chance also to remember that. And some on this call, on this Zoom meeting might remember that. Okay, so the outline for the talk goes like this. I'm going to start with a few remarks that are sort of tied to science about George. And then there are a few more scattered throughout the talk. For those paying attention, there's one quiz halfway through the talk. And then there'll be two fluid mechanical pieces, something on self-similarity and a feature of fluid mechanics intersecting biology. So some of these problems have overlap with talks by Ray Goldstein and Detlef Losa that were beautiful talks Monday and Tuesday. Okay, so where to begin with George Batchelor? Some on the talk, some on this call will have met him, some wouldn't have. So when I thought about this, I thought, well, George was precise, he was disciplined, he was organized. Patrick Aware told us yesterday about George's role in the co-founding and leading of Euromech. And I think it's fair to say that George worked to instill these attitudes and skills in the community. Now, here's one example that illustrates that he was not shy about expressing his opinions. In the volume 2 of the Journal of Fluid Mechanics in the late 1950s, he reviewed a book in the area of turbulence. He was not pleased with the book. His book review was 4 pages in length. The first 2 pages are what a book should accomplish, and the last 2 pages were his opinions about the book, including elements of the writing style. So now is a story that I tell every year when I teach fluid mechanics, when I come to the continuity equation. Here's what George wrote. And I should say, I think this is the next to last paragraph of the 4-page review. He writes, such a contribution as the book does make, and it cannot be reckoned as large. Oh, that was in the middle of the review. And then the next to last paragraph is, it is of course always possible to learn something from a new book. I myself picked up one little wrinkle, which I have stored away mentally for use on occasions when I wish to impress students with my progressive outlook. It is that when one is establishing the equations of continuity, it is nowadays desirable to exclude the possibility that nuclear reactions are going on in the volume element under consideration. That was his one feature of the book he was willing to acknowledge. Okay, I asked my colleague and advisor, Gary Leal, who worked with George for a couple of remarks. This was more than a year ago, when we thought the meeting would be held in March of 2020. Gary remarked that he was the first chemical engineering student to work with George. And he recalled that George remarked that he wasn't sure whether Gary was the better applied mathematician or George was the better chemical engineer. Gary took this as a compliment, but admits the remark could have been interpreted differently, which I thought was rather amusing, so I thought I'd share with you. And then Gary commented, which I think several of the previous speakers have indicated, that when you talked to George, it was important to know what you were talking about. If there was uncertainty in your mind, it would come out instantaneously through George's penetrating questions. And Gary remarked that in a paper he co-authored with John Hinch on the Brownian contributions to the bulk stress in a suspension, the previous speaker even mentioned some of George's work on suspension mechanics. In that paper of Hinch and Leal, George refused to believe that there was any contribution to the stress that could not be calculated via the deterministic hydrodynamic disturbance flow. He became convinced, or at least was sufficiently unsure of his position, that he eventually allowed John and Gary's paper to appear in JFM. So let me move forward. So it's been commented already that George had several sort of distinct areas of his career. I just broke them down this way. Turbulence, we've heard some beautiful talks, 3 beautiful talks the last 3 days in their turbulence. His magnificent textbook, An Introduction to Fluid Dynamics, and the 3rd stage of his career in low Reynolds number hydrodynamics. But I want to remark that I think those who knew him would say that he had a lifetime of careful listening and he made insightful suggestions. And I have one story I wish to share on the insightful suggestions. But if you want afterwards, we can talk about other places in published articles of others where his insights are acknowledged. So this is from Andy Acrivos, already also something I collected a year ago. And it's a remark Andy made where George's contributions to an important area of suspension mechanics may not even be appreciated. And so I'm just going to read this story. Over 40 years ago, my PhD student, Francis Gadala Maria, a Princeton graduate, carried out experiments on the rheology of concentrated non-Brownian suspensions, the results of which had us scratching our heads because they went counter to what we were expecting. Francis graduated and was succeeded by Dave Layton, another Princeton graduate, whose task was to make some sense out of Francis's experiments. But we were unable to make any headway until just before Christmas 1982, I had a long discussion with George Batchelor, who shortly thereafter suggested that Francis's observations were due to the phenomena of shear-induced particle migration discovered some years earlier by Eckstein, Bailey, and Shapiro at MIT. The field of suspension mechanics hasn't been the same since then. And the reference is at the bottom. Now, just to make clear what this communication was, this is the letter that was George Batchelor wrote to Andy Acrivos in December 1982. I have it here, I won't read it for you, but George, beginning remarks about your experiment, and it writes, I think it is likely that the further lifting of the bed of particles following the initial dilatational rise is due to the transverse convective diffusion of particles. He cites then this 1977 paper from the Shapiro group. He gives the typical, what's called now the shear-induced diffusion coefficient that depends on the shear rate gamma dot and the particle radius squared, and communicates now. I guess email is our current mode of communication, but this was George making an important contribution in a a wonderful way to another colleague, a very impactful colleague at that. Okay, so I'll leave you with that story. Okay, so those are a few remarks. I have a couple others later in the talk. And now I want to tell you a little about some fluid mechanics. And the first thing I'm going to tell you is work done by my graduate student, Nan Sue. A paper came out last fall, and it's about what I think is an unusual similarity solution. And at the end, I'll mention one or two others who had stumbled on a similar thing. And this will have both an experiment and a analytical feature. So I don't have to tell the listeners here about similarity, but you know, if you're trying to explain it to someone, you might start with geometric similarity. I share this with you because the two reviews at the bottom are probably not known to most of the people on this call. They're two beautiful discussions of self-similarity in fluid mechanics, one by Ivar Graton and one by Milton van Dijk. But the more common one is probably what many of you teach. And when you teach, say, heat conduction or chemical diffusion. You write down a diffusion equation shown on the slide for some, say, chemical concentration. And if you had a quantity diffusing, say, into a half space where the concentration was fixed at some number C0 at the wall, you might, before you actually do any math, sketch the figure on the left, which is, if you can see my cursor, a set of concentration distributions at different times. You might then try to convince the students by dimensional reasoning that these curves may look similar if you scale position x and time t as this so-called similarity variable on the right to get a universal curve for the concentration distribution. I'll just say that if you were trying to describe this to someone, you might say this is a partial differential equation in two variables, which you succeed in reducing to an ordinary differential equation in a single variable. Okay, so there are many variants of this, and the point I want to make is by far the great majority of problems, almost every example I know in the literature, are examples of the type where you often have diffusion-like equations, either the linear diffusion equation on the left or the nonlinear diffusion equation on the right, here written as a second-order nonlinear diffusion equation. And when you can find a similarity solution, it has the feature that it involves a variable, say eta, which is the ratio of the two independent variables in some functional form. And if you're able, you make this change of variables, you can generally, or for problems at hand, reduce the nonlinear PDE to a nonlinear ordinary differential equation. You need boundary conditions, of course. You hope you can solve it. If not analytically, then numerically, and then you want to compare with experiments. That's a standard procedure that some people have done, some of you teach, and we're happy when we find it. So the question I want to ask, though, is about systems that involve three variables. And is it possible to find a similarity solution? And by this, I mean where you reduce the problem all the way to a nonlinear ordinary differential equation. And if you do it, can you give me some examples? I'll show you one of ours and two of another group. And then can you show experiments? And I only know the experiments that we've done. So that's this intermediate theme. Now, from three variables to one variable, I'll point out one example I know that sort of has this feature. It's a slightly different kind of problem, but many of you may teach the G.I. Taylor scraper problem, where you have a corner flow that's driven by a moving boundary, here the bottom boundary relative to a fixed upper wall. This corner flow has a very nice, of course, analytical solution. And a three-dimensional variant of this was given by Keith Moffett and one of his graduate students. And that problem has the feature that you have an inclined fixed wall and a rotating bottom wall. In the coordinate system shown on the slide, you use a cylindrical coordinate system here labeled r theta z, and it turns out the stream function for the low Reynolds number flow problem, if you solve it, is actually the rotation rate omega times z times r times a function of angle. And it turns out the function of angle is exactly the functional form discovered by G.I. Taylor for this two-dimensional flow. So this would be an example, I might say, of a problem that lacks a length scale. So it yields to a self-similar solution. It involves all three independent variables. Of course, this is for a linear problem. The fourth order differential equation that describes the stream function. Okay, so I want to tell you about thin films, and I'm going to remind you about a classic problem first studied by Harold Jeffreys, which is the drainage of a film on a vertical plate. The film is shown here labeled colored in blue, and X is the direction in the direction of gravity. This was studied about 90 years ago by Harold Jeffreys, and he first derived a differential equation for the height h as a function of x and t. It's driven by gravity, and mu here is the viscosity. And Jeffreys noted the exact similarity solution, which I'm going to denote for the rest of this talk as h with a subscript j, as the square root of the viscosity times distance divided by rho gt. So it has this nice, clean, power law-like analytical solution. I'll remind you of this once or twice. But this is a classic thin film problem that comes up in a number, well, actually a large number of cases when gravity drives the flow. Okay, so this area of thin film fluid mechanics has lots of diffusion-like equations in it. And for those of you who haven't seen this, which I assume is some large number of people on the call, I thought I would just outline for you the kinds of problems that come up in the structure of the equation that is found. So if you want to solve the incompressible form of the Navier-Stokes equations, where mu is a constant, and you want to consider any geometry that's long and narrow. So here I've sketched different kinds of spreading films on a horizontal substrate, where h denotes the shape of the film. Then there's a classic nonlinear partial differential equation, for the shape of the film. I've written it here. I'm going to give you a brief derivation of it so you can appreciate how it arrives from Navier-Stokes. But it involves viscous effects through the viscosity times the time rates of change of height. It involves derivatives along the film. Here I've written the gradient operator with a subscript perpendicular, because it's perpendicular to the normal. So it's gradients along the film. It involves here a fourth order operator involving the surface tension, and two terms involving gravity, one involving two derivatives, and the last term is the Jeffrey term involving one derivative of the height function. So it involves, in this case, viscous surface tension gravity effects. If you want to know where this comes from the Navier-Stokes equations, I'll just say, if this was your configuration and you like lubrication theory, then you note that if the film shape changes slowly, then you can use lubrication. And lubrication means you can write a one-dimensional effective model for the average velocity for the flow along the film. And you need to find the velocity along the film, and then h is again the film height. And for the velocity along the film, you turn to Navier-Stokes. You neglect the inertial terms. You approximate, as in Darcy, the viscous term by mu velocity divided by h squared, the minus sign because there's friction. And if you substitute this result into the continuity equation and identify the pressure either with hydrostatic effects or surface tension effects, or this is popular because you can also include elastic effects. But if you have the former two, then you get this differential equation almost immediately. And it's a nonlinear diffusion-like equation, and it has many variants, of which I'll show you a new one today. Okay. So there's that part of my talk. Okay, so where do you go from here? Well, we were interested in experiments. We were actually doing some experiments on a wetting problem in the spirit of physical chemical hydrodynamics that Detlef told you so beautifully about yesterday, that if you dip a film into a bath of surfactant solution, you could see the film rising up the plate. Detlef told us yesterday about different kinds of Marangoni flows. But in doing these experiments, which always involved a draining film, one day we decided to do them sort of in reverse. Nansue just started to look at film drainage. And I told you that film drainage was first studied on a vertical plate by Harold Jeffreys 90 years ago. And showing on the slide is the movie that we made. I'm going to play it once more. And what you'll should notice right away is, and I'll try and convince you of this, that the middle of the film will look like the Jeffrey problem, but the most distinctive feature are the edges. And the edges show that the film thickness is different because the dye intensity is different. And we got very interested in these edge effects and decided to try to look at the so-called boundary layer that occurs in this problem during the film drainage. And I'm going to convince you that this is a 3D self-similar structure. So again, I'm going to show you some data of the middle of the film. To first show you the kind of things we did, we just collected light. So we measured the dye intensity. The picture on the bottom are 4 different images that show in the middle of the film the dye intensity is uniform, but that the structure that's formed near the edge, is this, what I'm going to refer to as the boundary layer. And we got then most interested in, could we quantify it both experimentally, and then what could we say about it mathematically? So we did interferometry. Interferometry allows you to determine the film thicknesses, or at least the changes in film thickness. So I don't know how well this is going to display over the internet all around the world, but an interferometric movie is playing on the screen right now. What you see as the spacing between the fringes tells you the change in the film thickness from place to place. And what you'll see, because the fringes are horizontal in the middle, that the film has a uniform thickness, and it changes as you go down the plate. And then near the edge, the film thickness is changing because the fringes are no longer horizontal, and their spacing is also changing. So we make very detailed measurements and can deduce film thickness. So what did we measure? Well, the first thing we did is we wanted to just check the Jeffrey solution, which said that the film thickness should vary as the square root of x over t. So at 3 different places, we measured film thickness, and we did it for 2 different viscosities. So we have 6 different data curves here. And Jeffrey's claims that the solution should be self-similar. So if you scale the height by the Jeffrey solution, we find a universal curve. And that just confirms what the experts would say they already knew, which was that the Jeffrey solution provides a clean, analytic, quantitative description of film drainage. Okay, so our interest though was the edge. So we started to look near the edge. And so we picked one position, X, and then we scanned Y, which is the horizontal dimension. So here is film shape H versus Y, the transverse coordinate for given X. We did this at 3 different X locations, and we did it for 3 different times and 2 different viscosities. And so you see you have all these different solution curves. And the solution now involves 3 variables, X, Y, and T. So we wanted to ask, in what sense could we understand this? And so here's the feature that I'm now going to tell you about that's quantitative. And there's always also a location where the film height has a maximum. That's this dotted blue line, and that makes the whole thing sort of look like your classic boundary layer. So here's the nonlinear PDE I told you about to start with. This is in the literature. It's very well known. And it's been studied in many variants. Okay, so the first thing is we're studying a vertical plate, so one of the gravity terms goes away. Second of all, we're going to make a boundary layer-like assumption. I've sort of sketched a picture of what we imagine the film profile to look like as a function of X, Y, and the vertical position. But we assume that the variations along the flow, which is the X direction, are much slower than the variations perpendicular to the flow. And the consequence of that is that the surface tension term just becomes a 4th derivative term in the Y direction. And therefore, overall, the thin film equation becomes this nonlinear PDE involving time variations, which are viscous effects, surface tension effects, which involve the 4th order operator, and the gravity term. And the first and last term are the Jeffrey solution. Now, I'm just going to sort of tell you what the solution is we stumbled on, but you can do a proper boundary layer calculation to show the same thing. So what we noticed when we first started to look at this was we sort of first guessed that the solution far away is a Jeffrey solution is because of surface tension tied to the shape of the film. So we looked for a solution to the nonlinear PDE of the form, the Jeffrey solution, which is a function of X and T, times a function of a single variable eta, where we sought eta as a function of all 3 independent variables. And sort of miraculously, or maybe not miraculously, depending on your point of view, but when we did it, we thought it was sort of miraculous. This similarity variable involves time, x and y, all three of the independent variables, and it reduces the nonlinear PDE to a nonlinear ODE that you can solve numerically, and or also if you linearize, you can find an analytical solution with the help of special functions and Mathematica. And so going back to what I showed you before, we had this data at a given position, x, where we plot film height versus y. We do that at different x, and we do it for different times and different viscosities. And then we ask, how well is the similarity solution that I just posited for you, where f of eta is a known solution to an ordinary differential equation. And so you can try to collapse the solution h by the Jeffreys solution, and you get a function that roughly collapses to a single curve as a function of this similarity variable. And if you take either the numerical solution to the ODE, or in this case, I've just plotted the linearized solution, it does a remarkably good feature of capturing the shape of this curve. Now, you might wonder about different quantitative features because the similarity variable involves different exponents. And so you might ask, how well does this kind of feature work? So we took the film peak. Every measurement had a peak height, the largest peak height, which sort of is indicative of the boundary layer thickness. And then we plotted, for example, what the film height is. And so here's our theory. And we first said the peak height known numerically is this value of eta. And then we looked for what the distance to the peak height is, y, as a function of either x or t. If you do that, it looks like this. If you plot the peak height y versus time for different x's and 5 different viscosities, you get all the different data shown on this graph. This is sort of the boundary layer thickness changing in time as drainage happens. But if you collapse it according to the similarity variable, you get a reasonable collapse. There's some spread, but any given data set is very close to time to the -1/8, which is predicted by the similarity solution. And if you do the same thing the other way, but look for shape as a function of position at a given time, so you want to know the spatial structure of the boundary layer, you can plot the thickness of the boundary layer versus thickness versus x at different times. Here are 3 different times and 5 different viscosities. And again, if you collapse this using the similarity solution, you get now a tighter collapse with respect to the spatial variable, and very close, the x to the 3/8, which is predicted analytically, is very, very close to what we measure in the lab. And so this kind of concludes this first part of the talk, and is the kind of suggestion that here's an illustration of a nonlinear PDE in 3 variables, which can be collapsed from 3 variables to 1. I've shown you just the sketch of the problem, but if you want a proper boundary layer like solution, you can do that on this equation. And in that sense, then there's a natural small variable, which is the bond number. Detlef talked about that yesterday at length, which is the ratio of the capillary length of the fluid, in this case, relative to the width of the plate. And if that bond number is small, then you have a small parameter that naturally gives you a boundary layer structure. Okay, so that's my first short story. Okay, well, I can't ask for questions because we're on this form of a presentation, so I'll go on with my talk. There's an important thing to say. After we published this, Stephen Wilson shared with me a very beautiful paper that he had written already 10 years before with a feature of this idea, which is in a purely gravity-dominated case, they found an example mathematically in joint work with Brian Duffy and colleagues on a gravity-driven problem, which had what I would call the collapse of 3 variables to 1 variable. And just a year ago, his former student Yitam, I'm sorry, I've misspelled it here in the second reference, has a mathematical solution of effectively the same kind of problem that we described. It's surface tension dominated, and it's a drainage problem. where he also shows this collapse of 3 variables to 1 variable. So there are 2 mathematical papers out there that also have this very nice idea that I've shared with you. Okay, so at this point in the talk, you might be wondering, what do you think about next? So I think about something that I call good versus bad physical arguments. It's a remark that George Batchelor made, and I learned about this from a paper co-authored by Andy Acrivos, David Jeffries, and the late Dudley Seville, whose pictures are shown on the right. And I'll just read to you. This was at the end of a paper they wrote about 1990. They wrote, they had a very mathematical paper, by the way, but they tried to explain where things came from. So they wrote, the references in the text acknowledge formally the influence Professor Batchelor had on this work, but less formally, the interplay between physical and mathematical arguments discussed in the paper brings to mind of one of us, David Jeffries, an exchange that took place during a seminar at Cambridge in the mid-1970s. So for those in the audience that know the famous Friday afternoon seminar at DAMPT, you could imagine the following happening at the end of a seminar late on a Friday afternoon after the speaker finishes talking. The speaker, trying to justify an elaborate mathematical argument, states, the trouble with a physical argument is that you may not get all the terms. George Batchelor was in the audience, remarked, no, that is the trouble with a bad physical argument. Several voices are heard in the audience. How do you tell a good physical argument from a bad one? George Batchelor, that's easy, you think. Okay, so we always want to do our best to think carefully and do our best to give clear arguments. Okay, so here's your trivia question. Oh, it's not animated quite right. So the trivia question, I guess I didn't animate my slide properly. The trivia question is, how many papers in Nature did George Batchelor write? And, for some, they view Nature as a very prestigious place. And so that would be a quite prestigious place. And maybe you don't associate George with writing to Nature. And I didn't animate this properly. I gave you 3 choices, but I've actually put the answer on the screen. So that was bad on my part. But in fact, he wrote 3, had 3 publications in Nature. I'm going to guess that most people in the audience, even those who knew George, didn't know that. 2 of them are about science, papers in 1945 and 1946. And I encourage you to look at them if you'd like. The last in 1982 is about the political arrest of a fluid mechanicist, Ryszard Herzinski, in Poland during the government's crackdown, and even on scientists who were speaking out against politicization of science. Professor Herzinski's son, Andrei Herzinski, may actually be on the call, is also a distinguished fluid mechanician at Boston College. Okay, so, and I'll have more to say maybe about the theme in a second. So I thought you might find that interesting. For those even who knew George, they might not have been aware of this. Okay, so George's second career was in microhydrodynamics, what some people will call low Reynolds number flows, but I just want to comment about a very beautiful feature of it because it resonates with several of the talks in this special program. In the proceedings of the IUTAM in 1977, he wrote a beautiful article. It's referenced here. It's called Developments in Microhydrodynamics. And let me just give you George's words because it really goes far. It says, microhydrodynamics deals with processes occurring in fluid flow when the characteristic length of the flow field is on the order of 1 micron. I think that's too restrictive, but ignoring that for a moment. This paper reviews the characteristic effects of microhydrodynamics and investigates it in reference to Brownian motion, particles in moving fluids, and the collective properties of suspensions. He then goes on to add, attention is also given to flexible long-chain polymer macromolecules, the beating of flagella and cilia, nicely illustrated in Ray Goldstein's beautiful talk on Monday, and electrokinetic phenomena in the framework of microhydrodynamics. You could have also probably added to that physical chemical hydrodynamics in the spirit that we heard about yesterday from Detlef Loser. Okay, so with that, I will go to my second fluid mechanical story, which is a study of a biology problem that relates to a fluid mechanics problem. And I believe it may be very important in pointing out to some of you another place that fluid mechanics plays a role in molecular biology, in an area of molecular biology that's just now being discovered. Okay, so there are little short stories here. I just get to remind you of the so-called Rayleigh-Plateau instability, which involves surface tension. Here, I'm going to apply it at the scale of nanometers. So if you wonder about scale where you can apply continuum mechanics, here you'll see a nice, at least relatively small scale. I'll point out to you, for those, I'm going to guess there are many fluid mechanicians who haven't heard of this, but a new discovery, new recognition in molecular biology called liquid-liquid condensates. I'll remind you of something you learned in biology called how the spindle is formed, and then I'm going to put them all together to show how fluid mechanics helps play a role, previously unrecognized role in this process. And a feature, well, I should also call out, involves a form of nucleation of the spindle called branching nucleation that my colleague Sabina Petri in molecular biology at Princeton is one of the pioneers. Okay, so you're probably, almost everyone on this call is probably familiar with drops breaking up by surface tension. We saw a little of that yesterday. And if when you first study this problem, as first studied by Lord Rayleigh, and theoretically and Plateau experimentally, these drops break with a certain wavelength. But there's a variant of the problem that is shown on the bottom of the slide, which is a thread of liquid coating a wire. This was first studied 60 years ago by Simon Gorin in a beautiful JFM paper. And there you study the Rayleigh instability, but with an inner boundary. And the important parameter is the film thickness relative to the radius of the wire. In all of these problems, I should say that there's a typical speed, which is the surface tension divided by the viscosity. Here I've denoted the viscosity by eta. And there's also a typical time scale, which is the radius of the fiber divided by the typical speed. That's important because in all these problems, it turns out the viscosity, the surface tension divided by the viscosity appears as a parameter. So it's very difficult to get either, but you can get their ratio. Now, if you took a biology class a long time ago, or even in college, what you typically have learned is that organelles inside the cell are separated from the material in the cell by membranes. But what was recognized about 10 years ago, and apparently there are precursors going back several decades before that, is that there's many structures inside the cell that don't have membranes, but they functionally organize materials by phase separating. And I'm playing a few movies here of this, where you see that the materials are flowing like liquids. They even form small droplets. And that was the evidence that this phase separation is producing liquid-like structures that are likely bound by surface tension, and they flow like liquids, which means they have a viscosity. And this has now been found all over molecular biology. The references here are from the last 12 years, and it's a paradigm that is suggesting to people other places that liquid-like phenomena occur inside cells, although not with any membranes. Now, so that's about liquid phase separation. The other thing I have to remind you of is you probably once learned about the spindle that's formed inside a cell that allows you to separate the chromosomes every time a cell divides. And a famous, popular movie that you would have seen of this is being shown on the slide, where you see the spindle form, and then the mechanics of the spindle allow you to pull apart chromosomes. In Sabina Petri's lab, Akanksha Thawani, who is a joint graduate student with Sabina and I, did, with help from Josh Shavitz in physics at Princeton, did beautiful imaging to show how the spindle is being constructed in reconstituted materials. And here you see the fact that as the spindle forms, you can quantitatively see that it's filaments that are often branching off of other filaments in order to span a certain angle as the spindle is growing out. This structure is called branching nucleation. And it turns out it's known that an important factor in this branching nucleation process is a protein. The protein has a name called TPX2. And I'm going to tell you in a second that TPX2 actually forms liquid-like phases that exert surface tension, and then plays a fluid mechanical role. And I'm going to document that for you on the next slide. But this is the last piece that now shows that in the understanding of the spindle formation, it's important to understand how the spindle gets constructed and what are its mechanics. And I'm next going to tell you that in there is a fluid mechanics-like problem involving microhydrodynamics. Now, I should say, over the last 6 years, there are a lot of images in many papers all over the literature documenting filaments, either so-called microtubules or other filament-like structures that have liquids on them. And they often see the liquids, I don't know how well this will show up on your screen, they often see the liquids forming small droplets on fiber-like features. You can see it in a number of these. Now, each of these different images has a different protein, which means a different condensed liquid phase. One here is called bug Z, another is called TAW, and on the lower left is the TPX2, what I told you about. So this is just meant to emphasize that in the area of molecular biology, there are many proteins. They form liquid-like phases. They often form droplets, but no one had ever made a remark in this literature about a functional role for the droplets, okay? And no one had ever said, why do droplets form, how big they are, where, why might they be there? And so that's something that we stumbled on. And in work from Sabina Petri's lab a year ago, they had noticed in one of their experiments that what had appeared happened, and I've highlighted in yellow, that the microtubules that were forming the spindle and allowing branch and nucleation to form had regularly spaced patches of this protein TPX2 on it. And they even showed images where shown on the left here in blue, if it's showing up on your computer, is the protein TPX2. Labeled in red are the microtubules, and they saw branching coming on the right from a mother microtubule, and the branches seemed to only be coming from where there were little droplets of TPX2. When they do electron microscopy, it's known that you need several, and there's a little math problem here as well, you need several factors to co-localize to cause this nucleation. And what they noticed was that the factors tended to be sort of spaced in some sense along a microtubule. And I'll just point out that the microtubule diameter here is 25 nanometers. So you're looking at radii of on the order of 10 nanometers for the fluid mechanicals problem I'm going to describe next. We then did experiments where we pipetted in TPX2. This is work done by Bernardo Gavia and Segar Setru. And these images make clear that what you see is a filament first labeled in red. It has a coating that's formed on it, and the coating then breaks up in a series of droplets, which is well known to fluid mechanicians that think about the Rayleigh-Plateau instability. This happens in a wide number of experiments we've done. Always was leading to this form. And I should just say the film forms quickly, and then the drop breakup is very slow. So that's a feature of this kind of problem. We did it at physiological concentrations of TPX2, so we couldn't be accused of doing it outside the regime of where biology operates. And in every case, we get microtubules that are quickly coated with a film of TPX2. It forms a film, and that film then goes unstable to form a set of droplets. And the argument we're going to make is those droplets are actually playing a functional role in co-localizing factors that leads to this so-called nucleation process important to the spindle. Now, I mentioned this 1962 paper by Simon Gorin. If you want a nice low Reynolds number flow problem, Gorin calculated the calculated the growth rate for any given wavelength or wave number. And so that only depends on a single variable, which is the film thickness, here denoted by H, or the outer radius R naught, relative to the inner radius. And so you can plot this stability diagram as growth rate versus wave number. The only place for surface tension and viscosity is setting the growth rate. The peak wave number is only a function of the aspect ratio of the problem, so the film thickness. And the film thickness, we're always controlling in experiments by how much protein we add to the solution. So there's a well-defined growth rate, and it varies with film thickness. And we tested that experimentally by doing atomic force microscopy, where we coated the system. We then measured the film thickness at different times, and we watched a system that shown here in the upper right has the film height being effectively uniform at 25 nanometers. That's the bare microtubule. After pipetting in TPX2, which forms this liquid condensate on the surface, you get the blue curve, which is at around 40 nanometers in thickness. And then over time, a periodic, nearly periodic modulation occurs. You can do a Fourier analysis on this and pull out the wavelength of the fastest growth rate. And you can, sorry about that. And you can do this then for many different film thicknesses and many different microtubules. And so we did this, well, Sagar and Bernardo did this for a large number of systems, and in every case could determine a most unstable wavelength for the system. And so here's a graph of film thickness on the horizontal, controlled by how much protein we add to solution, the measured drop spacing. The blue line is the theory of Gorin. The gray shaded area is plus or minus 10% to give you a typical idea of what you'd expect because of variables you are somewhat uncertain about. And 4 different data sets are shown here, which agree remarkably well with a fluid mechanical theory at the scale of tens of nanometers, using a classic result, which is just that there's a well-defined surface tension between the condensate, here the protein TPX2, and the outer solution. And this has no fitting parameters in it, and we argue is the reason for branching nucleation. And Sabina's lab has then went away to try to show that, and they did an experiment where they showed, I think I have a movie here, where that wherever you see branches, you almost always, if not always, see small droplets on the surface of a mother microtubule, and the branches emanate from these droplets, which we're going to say are the result of the Rayleigh capillary instability. In the interest of time, I'll just then say there's one more short story here, which is you can ask, what is the functional role, and why are droplets better than a thin film? And so we went to study that using a mathematical model that is familiar to some of the audience, looking at kinetics, and that is asking, are you better off with a uniform film if you're trying to co-localize multiple factors, or are you better with breaking the film into droplets? And what we discover by a kinetic simulation is that the time to co-localize multiple factors is always shorter if you're starting with them in a, if you like, a smaller volume, which is the volume of a little droplet, than being distributed everywhere along the microtubule. This was done by simulation, not with a analytical theory. And so our argument then is, what is the functional role? The functional role is to decrease the time to give branches, make branching more efficient. That's important then in spindle formation. And so this is actually one of the first examples that we're aware of a functional role for liquid-liquid phase separation inside the cell. And there are a number of other processes that are now thought also to be functional. And so I'll leave you with that. Okay, so I've gotten to the end of my talk. I've tried to have a couple features here. I tried to get you to think about George Batchelor and the importance of clear communication. I hope I've been relatively clear here in 2 problems. One is this unusual, at least I think unusual, edge drainage problem, a boundary layer like problem involving 3 variables, but you can get to a single ODE, and it isn't, the quantitative features are in good agreement with the experiments. And then this classic fluid mechanics problem that involves microhydrodynamics, if you like, involves physical chemical hydrodynamics, involves all kinds of stuff, involves biology, which is a small scale version of the Rayleigh-Plateau instability. Okay, with that, I just have one last thing I'd like to share. We live in times that sometimes seem more troubled than we would think they should be. And so I'll share with you one last piece from George Batchelor. And I think this is important, certainly because the last year has not been simple, not only because of the pandemic, but we've seen many different features of our countries not function as well as we might like. And the people on this call, we share something in common. We're from all over the world, but we share a love and a trust in each other in the science that we do. So this is George Batchelor, sort of near the end of his career, thinking about science as a civilizing and ennobling activity. He wrote, through having common objectives and principles by which new knowledge is assessed and disseminated, scientists concerned with a particular field like fluid mechanics form an international community of great unity and moral strength. I believe that the understanding, trust, and goodwill between members of the scientific community transcends geographical and political boundaries and constitutes one of the most important forces for international harmony and friendship in the world today. And I think many, if not everyone on the call, will understand that these words mean as much, if not more now than when George wrote them. And with that, I'll stop. Thank you.
Thank you, Howard. I agree entirely with George. One always has to agree with George, but one does agree with George on that international significance of scientists. Is the breakup only because of the instability, or are there do the ligaments flap around?
So I think you're asking about the second part of the talk where we suggest the Rayleigh-Plateau instability. There are lots of different sources of perturbations in these problems. At the scale of the cell, you're probably aware that the spindle is built by molecular motors, so there are many sources of fluctuations. The time scale of the instability, though, is slow. It occurs over minutes, so it's on time scales much long, well, many tens of seconds, if not minutes, on time scales much longer than the time scales associated with the fluctuations. And so our argument is that what you're seeing there for the typical spacing between droplets aligned along a filament is surface tension. And I think the argument we've given is the theory of the Rayleigh-Plateau instability is in very good agreement with the wavelength you measure. So we don't need to call on any other effects, but you need a perturbation, but there are many sources of perturbations.
So jumping to another question is, are there thermal effects impacting instability? But you said there were molecular motors, which will be much bigger disturbances.
Yeah, we don't believe that there are thermal effects. I mean, I know there's some discussion in the literature about ATP and small thermal effects that are occurring. We don't believe, at least I don't believe, that they play any role in what we're seeing here. And yeah, so I'll just leave it at that, Jonas.
Another question on these Rayleigh-Plateau droplets is from John Chapman. You've got these droplets on your main tube. Why aren't they on the branches as well?
Yeah, so the here the feature is if I understand John's question, the first thing we always see is that after pipetting in a solution, the TPX2 phase separates. So most of this protein first coats the available microtubules. That film then breaks up into small droplets, and those droplets then are all along a mother microtubule, and it's from those droplets that you start to produce new branches. Of course, those new branches then, if there's sufficient TPX2 around, will have a film of TPX2 on it. We haven't studied that in detail. Okay, so but drops are never migrating. The phase separation here is happening rather than in bulk, which is what was true in all other papers. Here it's happening on a substrate.
I think actually there is a slow sort of Oswald ripening where a bigger drop will grab stuff from nearby, but it takes ages.
Yeah.
There's a question about the first part, the drainage. Why is there a peak at the edges? Is it due to capillary activity?
Yeah, so maybe I didn't make that clear. The edge effect is a surface tension effect. So it's a capillary driven flow that is acting transverse to the direction of gravitational drainage.
Yeah, and it's driven by some contact angle condition on the edge itself.
You can put in different contact angles. We assumed, consistent with our experiments, that the liquids wetted the plate, and so effectively the contact angle was 0. But you could try to put in another angle if you want. I've tried to spend my whole career staying away from the contact line question, but here it was really a wetting fluid wetting the plate.